Everyone knows what the Black Death was. It arrived in Sicily in October of 1347 on 12 Genoese trading ships. The sailors were dead or dying, their bodies covered in black swellings at the armpits and groins. The Sicilian authorities ordered the ships out of harbor immediately, but the damage was already done. Within months, plague had crossed into mainland Italy. Within 3 years, it had swept through France, Germany, England, Scandinavia, and the Iberian Peninsula. By 1353, somewhere between 75 and 200 million people were dead. Between 30 and 60% of the entire European population had been wiped out. Some villages lost everyone. Some cities lost half. The scale of death was so catastrophic, it reshaped European civilization. Labor shortages that broke the feudal system. Theological crises that fractured the church. Social disruptions that, some scholars argue, accelerated the Renaissance. The story everyone knows ends there. The Black Death was a catastrophe. It killed, and then it was over. That is the story. And it is radically incomplete, because in 2022, a team of researchers published a study in Nature that revealed something the historical record had never captured. The Black Death was not simply a killing event. It was the most powerful evolutionary pressure ever recorded in human history. The people who survived it were not the same, genetically, as the people who lived before it. The plague did not just kill the unlucky. It selected the lucky, and the genetic signature of what it selected for is still running in the immune systems of Europeans today, not as history, not as ancestry, but as active biological machinery in living bodies right now. When scientists examined ancient DNA from before and after the Black Death, they found that certain immune gene variants had gone from rare to common in a single generation. The bacterium had run a massive, involuntary human genetics experiment, and the results were written in the bones of the dead. But the story gets darker before it gets stranger, because some of those same immune variants that protected medieval survivors from plague are now implicated in modern autoimmune diseases. The Black Death saved their lives. It may be contributing to ours. This is not a story about luck or divine providence, or the randomness of who the plague chose. This is a story about genes, and about what happens when a pathogen kills selectively enough to permanently shift the genetic composition of every population it touches. The evidence was sitting in ancient cemeteries, waiting 700 years for a technology to read it. The technology is ancient DNA sequencing, extracting and reading genetic material from medieval bones, and what it found in the cemeteries of London and Denmark reframes everything. It came from teeth and bone fragments, sealed in London clay for 700 years. Each sample was a few milligrams of material, enough, with modern technology, to read a genome. The fact that it worked at all is the technical miracle. What it found is the scientific one. The study that changed everything was led by Luis Barrero at the University of Chicago and Jennifer Klunk, then at McMaster University, with collaborators across Europe. It was published in Nature in October 2022 under the title, "Evolution of Immune Genes is Associated with the Black Death." What made this study different was the sample design. Researchers had previously extracted DNA from plague victims, the people who died. But to understand natural selection, you need to compare the dead to the living, or, in this case, populations before the plague to populations after it, and then compare those to people alive today. The team obtained bone and tooth samples from 3 cemeteries in London and 2 in Denmark, covering the period from roughly 1000 CE to 1500 CE. The London sites included East Smithfield Cemetery, an emergency plague burial ground opened in 1348. Specifically for Black Death victims, and a second pre-plague cemetery, providing a genetic baseline for before the outbreak. The Danish sites added geographic breadth and additional comparison points. From these samples, the researchers extracted ancient DNA and focused their attention on a specific region of the genome, the immune gene cluster, not the whole genome, not ancestry markers. They were looking specifically at genes involved in immune response, the regions most likely to show signatures of selection if plague had favored certain variants. What they found required them to sequence more carefully, and then again, and then verify against modern populations. Because the signal was strong enough to be immediately visible, and strong enough to be alarming, certain immune gene variants were significantly more common in people buried after 1347 than in people buried before it. The plague had passed over Europe, and on the other side, The genetic composition had measurably shifted. This is where the CCR5 research of the 1990s failed. It compared modern populations to each other. The Planck study compared populations across the plague horizon. That distinction changes everything. It is the difference between asking where survivors ended up and watching who survived. And the watch started from bones. 700 years of bones, finally able to speak. No study before this had the timeline. They had the victims. They had the survivors. They had the living. Putting all three together in the same analysis. That was the methodological breakthrough. The gene that produced the clearest signal was ARAP2. ARAP2 encodes a protein called endoplasmic reticulum, aminopeptidase 2. Its job is to trim peptides, small fragments of proteins, into the right size to be presented to the immune system for recognition. When a cell is infected with a pathogen, it displays fragments of that pathogen on its surface. ARAP2 is part of the machinery that prepares those fragments. Get the trimming right, and the immune system recognizes the threat quickly. Get it wrong, and the pathogen hides longer, replicates more, and kills. ARAP2 comes in two major versions called haplotypes. Researchers labeled them A and B. In populations before 1347, both haplotypes were present in roughly equal frequencies. In populations after 1353, the survivors, haplotype B, had become dramatically more common. The team calculated the selection coefficient for haplotype. In evolutionary terms, a selection coefficient measures how much more likely a trait is to be passed on. A coefficient of 0.01, 1% per generation, is considered strong in most evolutionary contexts. The selection coefficient for ARAP2 haplotype during the Black Death period was approximately 0.40. This is an almost incomprehensible number in human evolutionary terms. Natural selection does not usually work this fast, or this visibly. The Black Death compressed what would normally take thousands of years into a single, catastrophic generation. The team's modeling showed that haplotype B carriers had approximately 40% higher survival odds. If you carried haplotype B, you were dramatically more likely to survive long enough to have children. If you carried haplotype A, you were dramatically more likely to die. In 1348, ARAP2 was not the only gene that showed the signal. The team also found significant selection at TCM2, a gene involved in pathogen detection and activation of the innate immune response. The rapid non-specific first response the body mounts when it encounters a foreign organism. And there was a third signal at a gene cluster called TLR1, TLR6, and TLR10. The toll-like receptor genes, toll-like receptors, sit on the surface of immune cells and function as pathogen sensors. They recognize molecular patterns associated with bacteria, viruses, and fungi, and trigger immune activation when a threat is detected. Variants in this cluster also shifted in frequency across the Black Death divide. Each of these genes does a specific job in the immune response to bacterial infection. Yersinia pestis, the bacterium that causes plague, is precisely the kind of threat these genes evolved to detect. The alignment between the biology of the pathogen and the genes under selection was not coincidental. The plague was selecting for exactly the immune architecture that would give survivors the best chance. When the team looked at modern populations, living Europeans today, they found that post-plague haplotype frequencies closely matched contemporary European immune genetics. The shift that happened in the Black Death is not buried in history. It is present in living European genomes. Every person of European descent alive today has immune gene frequencies that reflect the survivors of 1347 to 1353. The laboratory work that produced the ARAP2 finding involved a method called haplotype phasing. This technique determines which genetic variants appear together on the same chromosome. Immune genes often function through combinations of variants that work together, not through single mutations acting alone. When the team phased the ARAP2 region, they found that haplotype B is not a single mutation. It is a block of correlated variants inherited together, influencing how efficiently ARAP2 trims peptides. In laboratory experiments conducted as part of the 2022 study, cells expressing haplotype B ARAP2 were more effective at activating the immune response against Yersinia pestis than cells expressing haplotype. The functional difference between the two variants was directly measured in the laboratory, not just inferred from population genetics. Researchers also noted that haplotype B produces higher levels of ARAB2 protein in cells. More enzyme available means faster processing, which means faster immune recognition, which means faster pathogen clearance against a bacterium that can kill within days. This level of functional detail is rare in ancient DNA studies. Most population genetics work identifies statistical signals of selection without being able to pinpoint the specific mechanism by which the selected variant conferred advantage. The ARAB2 result is one of the cleanest examples in the literature of connecting a historical selection signal to a specific biological mechanism, verified experimentally. 3 independent selection signals, 3 genes, all involved in the same biological function. That convergence, 3 separate genes all pointing the same direction, is the statistical backbone of the study. The plague did not leave a fingerprint. It left a fist print, still visible 7 centuries later. The twist arrived when researchers looked more carefully at what haplotype B actually does today. ARAB2 was selected for its role in helping the immune system fight off bacterial pathogens, but ARAP2 is also involved in immune regulation, the mechanism by which the immune system restrains itself. 3 signals, 3 genes, 1 function. The statistical case is not ambiguous. The plague carved a signature into European DNA that 7 centuries of subsequent generations have faithfully preserved. And that signature is still there, in every European genome sequenced today. From attacking the body's own tissues, autoimmune diseases arise when this restraint fails. Conditions like Crohn's disease, rheumatoid arthritis, and lupus are characterized by an overactive immune system attacking the body instead of external pathogens. And ARAP2 haplotype B, the variant that protected medieval Europeans from plague, has been identified as a risk factor for several of these conditions in modern populations. The same variant that improved survival odds during the Black Death appears to be associated with elevated susceptibility to Crohn's disease and ankylosing spondylitis, a chronic inflammatory condition affecting the spine. This is not a simple trade-off story. It is more complicated than that, but the direction is striking. The immune variant that the plague selected for so powerfully, compressing thousands of years of evolutionary pressure into 1 generation, carried a cost that only becomes visible centuries later, when there is no longer a plague to justify the immune system's heightened aggression. The medieval survivors did not know they were being selected. The selection was happening at the level of who died and who reproduced, driven by a bacterium with no awareness of the consequences. What survived into the modern era was not just a genetic variant. It was a trade-off, coded into the genome by one of the worst catastrophes in recorded history, with implications for the chronic disease landscape of the 21st century. This is what natural selection looks like when it happens fast enough to observe. It does not produce optimal outcomes. It produces survival in the immediate context, with costs that may not manifest for generations. The immune system of a medieval plague survivor was calibrated for a world with Y. pestis in it. Strip that pathogen away, as modern sanitation effectively did, and the calibration becomes wrong. The immune system is running code written for a threat that no longer exists at the same scale. And when it cannot find that threat externally, it sometimes turns inward. That is Crohn's. That is ankylosing spondylitis. This is not a failure of the immune system. It is a success that outlasted its context. The 2022 Rewrote study was not the first to ask whether the Black Death left a genetic fingerprint, but it was the first to answer the question with rigor and sample size to be convincing. Earlier work had focused on a different gene entirely, CCR5, delta 32. CCR5 is a co-receptor used by HIV to enter human immune cells. A specific 32 base pair deletion called delta 32 renders the protein non-functional. People who carry 2 copies of this deletion are almost completely resistant to HIV infection. The immune variant that saved medieval lives is now written into the genetic architecture of modern disease. Selection does not know the future. It only knows what survives right now. In the 1990s, researchers noticed that CCR5 delta 32 was unusually common in European populations, present in about 10% of Northern Europeans, and almost absent in populations from sub-Saharan Africa, East Asia, and other regions. This geographic pattern strongly suggested the mutation had been selected by some pathogen specific to Europe. The most prominent hypothesis was the Black Death. If CCR5 delta 32 had protected against Yersinia pestis the way it protects against HIV, then the plague could explain why this mutation reached 10% frequency in European populations. It was a compelling story, and it was wrong, or at least insufficient. Subsequent studies found that CCR5 delta 32 was present in European populations well before the Black Death. The plague could not have created it from scratch, and more recent work suggests CCR5 is probably not the primary receptor Yersinia pestis uses to enter cells, making the mechanistic story weaker than the correlation alone suggested. The CCR5 hypothesis is now considered an oversimplification at best. It remains an example of how evolutionary reasoning can be compelling before the data is robust enough. What the 2022 study did, which the CCR5 research could not, was move beyond retrospective correlation by comparing ancient DNA from before and after a specific event with large enough sample sizes and focused on the immune gene regions most relevant to bacterial defense. It produced direct evidence of selection rather than inference from modern frequencies alone. The difference is between watching something happen across the archaeological record versus arriving after the fact and trying to reconstruct what occurred. The London samples were particularly valuable because East Smithfield had been dated with exceptional precision. Radiocarbon dating confirmed that individuals buried there died between 1348 and 1350. This gave researchers a fixed temporal anchor. The genome of someone in East Smithfield is, with high confidence, the genome of someone who died of plague. The pre-plague comparison samples came from St. Mary Spital, representing a London population from the 11th to 13th century, well before 1347. Comparing these two London populations gave a before and after snapshot within a single city. Adding the Danish samples expanded the comparison to a second geographic region. Finding consistent signals in both regions strengthened the case that selection was driven by the plague itself. The statistical approach the team used scanned across the entire immune gene region, testing thousands of variants simultaneously and correcting for the multiple comparison problem. The signals that survived this correction, ARAP2, TCOM2, and the TLR cluster, were not simply the strongest signals in an uncorrected analysis. They were the signals that held up under rigorous scientific scrutiny. The result is a study that most geneticists consider the strongest direct evidence. Yet, that epidemic disease has actively shaped human immune genetics at a detectable timescale, not over millions of years of background selection, but within a span of time covered by the historical record. Two independent research groups also approached the Black Death's genetic legacy from different angles. One examined Ashkenazi Jewish populations, which show distinctive immune gene patterns that some researchers have connected to repeated exposure to epidemic disease in medieval Europe. Another examined ancient Scandinavian DNA across a broader time window, finding immune gene frequency shifts consistent with multiple waves of epidemic selection beyond the Black Death. Together, these studies are building a picture of epidemic disease as a continuous shaping force, not a series of isolated disasters, but a permanent pressure on human immune genetics across centuries. The standard framing of the Black Death is the death toll. The numbers are staggering: 75 million, 100 million, perhaps 200 million dead. The scale of the killing is incomprehensible, and incomprehensibility is a useful proxy for significance. But the death toll framing misses what the DNA now shows. The Black Death was not only a demographic event, it was a genetic event. And genetic events echo forward in time in ways that demographic events cannot. A population that loses half its members recovers, given enough generations. The dead are replaced by births. The loss is real, but the underlying genetic composition can remain unchanged if who survives and who dies is random. The Black Death was not random. It killed differentially. The people who died were, on average, more likely to carry certain immune variants. The method that Planck and Barreiro developed will outlast any single result. Every future epidemic with an ancient DNA record can now be interrogated the same way. The people who survived were more likely to carry the variants that gave protection against Yersinia pestis. That non-random death is what natural selection is, and when it is intense enough and specific enough, it changes the genetic composition in a single generation. Every European alive today is descended from the people who had a slightly better chance of surviving. The immune gene frequencies that currently exist in European populations, the ones influencing susceptibility to Crohn's disease, rheumatoid arthritis, and other autoimmune conditions, are partly the product of that selection. The Black Death is not over in the sense of being biologically inert. It changed the genome, and the changed genome persists. The sentence to hold on to, the plague, ended in the 14th century, but the selection it imposed is running in living bodies today. The history books record who the plague killed. The DNA records who it spared, and the difference between those two records, one demographic, one genetic, is the entire story. Demographic history recovers. Genetic history accumulates. Every epidemic adds a layer. The Black Death added the thickest layer ever measured in a single event. The 2022 Rewrote study is a methodological landmark, but also a small window onto a larger question. Every major epidemic in human history killed differentially. None of them killed at random. Each one interacted with the immune genetics of the populations it swept through, and each one left a fingerprint on the genetic composition of who survived. Ancient DNA is now making it possible to look for those fingerprints directly across the archaeological record. For the first time, the Black Death produced a signal strong enough to be visible in the 2022 study. But the Justinianic plague of 541 CE also killed between 25 and 50 million people. The Antonine plague of the 2nd century CE swept through the Roman Empire. The plague of Athens in 430 BCE killed Pericles and perhaps a quarter of that city's population. Were these events also strong enough to leave detectable signatures in ancient immune genetics? This is not ancient history in the sense of being finished. It is ancient history still running in your blood, in your immune system. In the chronic disease landscape of the 21st century, we do not yet know. But what we can say is that the framework has been established. The 2022 study showed it is possible to look directly at immune gene frequency shifts across an epidemic horizon. The same method can now be applied to every epidemic for which sufficient ancient DNA exists. The deeper question is what this means for how we think about autoimmune disease. If the immune variants running in modern European populations are partly the product of selection by medieval pathogens, then the autoimmune disease burden in European populations is not a random failure of biology. It is a consequence of survival. The accumulated cost of every epidemic that killed differentially. Crohn's disease, rheumatoid arthritis, multiple sclerosis, ankylosing spondylitis. These are conditions that disproportionately affect populations of European descent. The reasons are complex and multiple, but one thread running through that complexity is the immune genetic legacy of centuries of epidemic selection, including the single most powerful selection event in recorded history. The Black Death killed half of Europe, but it also made the survivors, and the survivors made us. The researchers who worked on the 2022 study are already looking beyond it. Every pandemic in the ancient world left populations behind, and those populations left bones. Ancient DNA is now recovering what those bones carry. The record of who survived, written in sequence. The results will come, one study at a time, extending the picture the 2022 pascals per s tarted. Each future study will answer, did this epidemic also select for immune variants in the survivors? And if so, what variants? Are those variants still running in modern populations today? The answers will not all be as dramatic as the Black Death result, but some may be. You are the survivor of plagues you have never heard of. Your immune system was shaped by deaths that happened before your country existed. The Black Death is the one we can see clearly now. The others are coming into focus. If that does not change how you think about what your immune system is and where it came from, subscribe. There is more of this coming. The researchers who worked on the 2022 study are already looking beyond it. Every pandemic in the ancient world left populations behind, and those populations left bones. Ancient DNA is now recovering what those bones carry. The record of who survived, written in sequence. The Justinianic plague samples are being analyzed. The Roman period samples are being assembled. Each future study will answer, did this epidemic also select for immune variants in the survivors? The answers will not all be as dramatic as the Black Death result, but some may be. You are the survivor of plagues you have never heard of. Your immune system was shaped by deaths that happened before your country existed. The Black Death is the one we can see clearly now. The others are coming into focus.